Maybe in the interest of time, we should probably get started. I see about 100 participants have joined the call. So welcome to this joint webinar on maximizing on minimal data using pharmacometric model to estimate the probability of technical success. This is a joint webinar between International Society of Pharmacometrics and American Statistical Association, Biopharmaceutical section. So those of you who are not who do not know about SXP, SXP stands for Statistics and Pharmacometrics, is a special interest group that was chartered by both ASA and ISOP. It promotes collaboration between statisticians and pharmacometricians to learn and grow and develop approaches to model-informed drug development. So we have a website. I encourage folks to register, join the SXP group, and also visit our website because we have educational content around pharmacometrics for statisticians and statistics for pharmacometricians. And also we have topics on MBMA and PKPD programming. With that, it's my honor to also introduce now Ted Lustig, who is the ASA Biopharm chair, to speak a few words on the ASA Biopharm. Ted.
Great. Thank you very much, Ram. So the biopharmaceutical section started in '67. We are, as mentioned, one of the co-sponsors for this particular webinar. We've been a section as a whole since '81. The '67 is actually the interest group founding. One of the things we try to do is bring together people from diverse areas, from pharmaceuticals, from medical devices, biologics, that are interested in the application of stats to the development use of these drugs and devices for humans and animals. And one of the things that I'm trying to stress in my term as chair is this idea about more of an inclusive approach than an exclusive approach. Like, we don't advance if we talk about things we aren't, we grow if we talk about the extra things that we are. So one of the things I'm very happy to continue in this collaboration is our close work with the pharmacometric section activities that are going on here. And I do want to make a specific call out to Alan Hartford, a prior chair of the section, who really championed this idea and brought it forward and saw that we can have this productive collaboration partnership that we have today. Next slide, please. So as a whole, we have notably two particular section-sponsored conferences, the Non-Clinical Biostats Conference, as well as the Regulatory Industry Workshop. Some of this information can be found via amstat.org, for the website of the American Statistical Association. There are a number of great presentations on various topics, including pharmacometrics, that are available through these conferences. So I'd encourage you to look at those and see what you might be interested in pursuing. Next slide, please. The volunteers are certainly in main point of how we are productive within our section. We have multiple committees ranging from the Executive Committee, Awards Committee, Publications Committee, Funding Committees, Outreach and Collaboration Committee, and the Scientific Working Group Review Committee, as well as the individual working groups. So many different channels, avenues by which, should you choose, you can become more involved in the section beyond something like this, which we already offer, of simply learning from the work of others and advancing your own professional development. Next slide, please. Some of the working groups we have, in addition to the SXP, from which this webinar is, are working groups on everything from Alzheimer's disease, estimates in oncology, real-world evidence, safety, software engineering working group, Bayesian working groups, health technology assessment. So a number of different ways in which you could also choose to be involved in terms of learning about and advancing various methods that are relevant to us within this section. Next slide. Again, if you'd like to join, amstat.org website is a good place to get started for us as well as for others. You can also see our particular section webpage at the community pages within AMSTAT that you can get linked to from there. We also have updates available on LinkedIn from our ASA Biopharmaceutical page that provides updates to things such as this particular event. But again, I'm very happy with this productive collaboration we've been having that was started by several persons such as Alan and Ram and being pushed forward. And I'm really looking forward to hearing the presentation we had today, which I think will be of broad interest. Ram.
Thank you, Ted. So it's my great honor to introduce our speakers. So we have speakers are from Pfizer. We have John, who is an associate director in pharmacometrics and systems pharmacology. Before joining Pfizer, John completed his PhD in pharmaceutical sciences from University of North Carolina, Chapel Hill, as well as PharmD from University of Florida. He works primarily in inflammation and immunology therapeutic area, supporting both large and small molecule development at various clinical stages. His interests includes methodology, automation, facilitation, and complex indirect modeling. We also have Min, who joined Pfizer in 2001. She has worked in phase 1 through phase 4 clinical trials in therapeutic areas of cardiovascular disease, oncology, ophthalmology, and virology, and currently works on late-stage clinical development programs in inflammation and immunology. She has MS in statistics from Temple and PhD in health-related sciences from Virginia Commonwealth University. With that, speakers, take it away.
All right, thanks, Ram. I will transition over to the slide deck. All right. So the title of the talk, as was shared, is maximizing on minimal data, right? And that's sort of a key point here. And I feel like as a practice, stats, pharmacometrics, all about getting the most information out of samples of data. And one of those pieces of information that is the topic of what we're talking about, and very important here, is probability of technical success. So just a quick disclosure here, sorry. Right, so we are talking about a Pfizer product. Min and I, of course, are both from Pfizer, as was mentioned. And yeah, so make of that what you will. So a little background on the disease state that we're talking about for our study. It's dermatomyositis. So it's a rare disease, so it wouldn't surprise me if there's a lot of people in the audience who hadn't heard of it before. It is associated with skin and muscle manifestations, and it's also associated with an elevated interferon beta concentration. And when you break down adult dermatomyositis, or DM, you can break it down into two big groups. There's amyopathic, which is to say basically no muscle manifestations. And that's really about 20% of all DM patients. And then the rest can be, again, kind of broadly classified into classical DM, which is a mixture of muscle and skin manifestations. So because of these, various presentations, there are two very relevant clinical scores, especially in the setting of clinical trials. There's CDASI, which the full spelled out score for name is on there, but the first, the C stands for cutaneous. So as expected, this is validated primarily for skin manifestations. Overall, the severity of as judged by CDASI does correlate, interestingly, to interferon beta levels. But again, this is really focused on skin. A newer score is TIS, Total Improvement Score. That's more holistic. It measures both skin and muscle manifestations, and it measures improvement over time. So it's not really a single time point measure. So for those reasons, it being holistic and it being a measure of improvement over time, it's really considered, when seeking approval in this indication, regulators strongly prefer you use TIS. So the data that we had going into the work here was from a phase 2 trial of our anti-interferon beta monoclonal antibody, which is now called dazukibart, in adults with dermatomyositis. So that was that study. And I'm going to discuss more about the study and its findings, but I feel like it's definitely necessary to give a bit more background in TIS. So that is comprised of 6 core set measures. I won't go into all of them. You could see them on the screen. I also refer to these throughout the presentation and colloquially as subscores. But for each of these are graded, a score is assigned for their improvement based on the absolute percent improvement. And you can see there's different weighting for each of these subscores. But the two biggest contributors and the two most relevant contributors to points I mentioned in the previous slide are measures of muscle manifestation, which is captured primarily in adults through MMT, manual muscle testing. And you can see strong improvement in MMT could result in a lot of points towards the overall improvement score, which we could see here. And then for skin manifestations, those are measured partially through extramuscular activity, which is down here. Now, this also says disease activity score, but in no case is the extramuscular activity, or as we call it, extramuscular global assessment, it is not equal to CDASI. They are both just happen to measure one in part skin manifestations and the other one primarily skin manifestations. So you would expect some perhaps correlation, but they're not the same thing. And then another point is improvement in each of those subscores, of course, results in a certain amount of points towards improvement. But then when you sum up for the total improvement score, you can also categorize that into minimal, moderate, and major improvement. And what we were looking at powering the phase 3 study for as a primary, and obviously what our PTS is based on, is moderate improvement, which is the TIS greater than or equal to 40. Okay, so that is a little bit more background on TIS. Now, the study design is very relevant to the discussion of how we determined PTS. So this is a very interesting study design. It was adaptive, and there were several stages to that development. So it was adaptive over several stages. So the first stage was looking at the skin-predominant subjects. So this is very early on. This was before we decided to look at TIS. I think TIS was maybe still in development as this was being planned. And we looked at CDASI in these subjects because they were skin-predominant subjects. And we only looked at 2 dosing levels, that being 600 and placebo. And so this was only in 30 subjects, but we looked at but we had from these subjects their interferon beta concentrations, a gene signature specific for interferon beta, and then IP10, which is a nice biomarker that we found in stage 1. We also measured their CDASI, their physician global, and we had SF36, which is relevant to the modeling. What we found there were very promising results with the CDASI, and so we decided to add a new stage. And there were 2 versions of stage 2, but what they had in common was that we added another dosing level of 150 milligrams. So we had placebo, 150 milligrams, and 600 milligrams. A later amendment to stage 2 just happened to have a crossover, but it's not really relevant here. What is relevant is we had about 30 more subjects, and we added a patient global, but really, of course, what we were looking for here was some dose ranging data. Interestingly, at the time we were making decisions for end of phase 2, we did not have interferon beta concentrations measured for these stage 2 subjects. And it's not like they were measured where there was uncertainty to the data, but it was that we just didn't have it. It was missing. It wasn't assayed. So we didn't have it at that time. So then we kind of were worried because we didn't have any data in subjects with muscle manifestations. These are skin-predominant subjects, and we didn't have any TIS data. So we wanted to power our phase 3 and make our decisions around PTS around TIS, so we needed some TIS data. There was no way we could get TIS information from stages 1 and 2. So we added stage 3 in muscle-predominant subjects. We went back to just the 2 dosing levels because we didn't get much more information from the 150 milligram in stage 2, and we didn't have a lot of time to recruit very many patients. So we ended up with only N equals 18 subjects. We still did not have interferon beta concentrations for these subjects, and we otherwise had all the same measurements, and then we had TIS. So there are obviously challenges here, just raw statistically speaking, despite the fact that we had, nearly like 80 subjects worth of data, we only had TIS in a very small subgroup. And at the time of the decision, it was really just 9 and 9, because we were providing this analysis mainly at the sort of crossover point between the 600 milligram and placebo. And we had no information on TIS in skin-predominant subjects, so we could not incorporate this information. Pharmacometrically speaking, we didn't have interferon beta concentrations. So if we wanted to somehow link PK/PD to the exposure response, which would of course be a nice way to consider all of the data, you'd have to overcome that as a challenge. So those are the challenges, and we're going to kind of discuss how we overcame them. But then what are kind of the key results that we are working with, which of course have their own challenges associated with them. So all of these are from the clinicaltrials.gov key results. So you can check there and confirm. But so for stage 3, we had the mean TIS per arm at various sort of weeks along the process. And as you can see, we're just on the cusp of an average improvement in our 600 milligram group at very close to major improvement, which is good. But we also saw a very strong placebo response, which we expected maybe closer to what we saw at week 4 to week 12. So, it kind of brings to question, well, how do we interpret the 600 milligram data? And then, of course, it reduces the delta that we observed. So with 9 subjects per group, very big uncertainty bands. So it leaves a lot of kind of open questions. For CDASI, for the data was very similar for stages 1 and 2, but I'm just showing stage 1 because it's a little cleaner because we didn't have multiple kind of amended versions. But we had stage 1 subjects who were skin predominant. We did see a huge reduction from our drug compared to placebo, which we were very optimistic about. That's what led to the rest of the trial. And then for stage 3, where of course these were muscle predominant subjects, not as much room for improvement in their skin manifestations, they did see numerically a greater reduction in CDASI, but it's not necessarily relevant to the interpretation because we knew they were muscle predominant. So where it's relevant, of course, for stage 3 subjects is looking at their MMT, which of course we only had for stage 3 subjects because it's a TIS subscore. And there we could see 20-point improvement and 10-point improvement for 600 milligram and placebo subject respectively. And these numbers don't really provide a lot of context, but the take-home point is that we had a good muscle response in our subjects with muscle predominant disease. We had a good skin response in subjects with skin predominant disease, and our TIS response in, again, just muscle predominant subjects is good, but there's a lot of uncertainty there. And it's kind of hard to, figure out how to combine all of that information together. And then, so it gets us to what were the end of phase 2 and then additional planning for phase 3 problems at hand. So like I said, we had a very strong observed CDASI response, and we had what I would consider here a modest but very uncertain TIS response in stage 3. So the question is, what is our most likely effect size for TIS? So that is to say, we had an observed effect size, and then we had a very wide confidence interval. Where do we expect along that confidence interval is our true effect size or our population level effect size? And then what is the expected response in phase 3, which is to say, around that variability in the effect size, where do we kind of expect our response to be most likely? Combine these two questions, of course, you get what is the probability of technical success for the program. And on that note, before I hand it off, probability of technical success, the approaches that we used, and sort of a background for those who are less familiar with probability of technical success. I'm going to hand it off to Min to cover that background. Yeah, so Min, take it away.
Thank you, Zhang. So first, I would like to acknowledge that the contents on the next 4 slides on probability of success are synthesized from Pfizer internal presentations and workshops on this topic. Next slide, please. So first definition on 2 terminologies. The probability of success is defined as the probability of the next trial or trials being judged successful according to some criterion or criteria, typically on efficacy. So in other words, the POS represents the likelihood of the next trials meeting the predefined success criteria, typically focusing on efficacy. And then the next one is a probability of technical success, PTS, is defined as the probability of compound generating data to support target profile in order to file. Therefore, PTS envelopes POS while incorporating additional considerations and such as safety, other endpoints, immunogenicity, or multiple phase 3 studies. The calculated POS may be the upper bound of that assessment of PTS. And in some situations, the POS is essentially a PTS. Next slide, please. So conceptually, POS is calculated as the probability of the event R defining success integrated over the prior distribution for the treatment effect. That is taking account of uncertainty about the treatment effect. It is the Bayesian version of power, also termed as assurance. So it is an acknowledgement of the fluctuations and unpredictability inherent in clinical outcomes. The differences from the frequentist power calculation of a study will be described in a later slide. Next slide, please. The elements that contribute to the calculations of POS and PTS can encompass a vast array of evidence, including but not limited to previous experience with indication, previous experience with endpoint, previous experience with population, information from other drugs, information from placebo treatment, information from literature, and also willingness to make some assumptions. We need to assess what we know and what we need to know. What we know may be data on certain populations and endpoints under certain dosing regimens. We need to ask ourselves is how well this matches what we need to know. So to fill this gap, we will require other sources of information and assumptions to link them. And the considerations for building a predictive model include, but are not limited to, the endpoint to be used in the next trials, the patient population in the next trial, the dosing regimen in the next trial, and the design of the next trials. Please note, the appropriate predictive model may not be the model estimated in the primary analysis for the last trial. It could be a secondary or exploratory endpoint of the last trial, a composite measure of selected endpoints in the last trial, or an endpoint can be estimated, extrapolated, or derived from the outcome measures of the last trial. Next slide, please. You can keep clicking. Yeah, thank you. So the main difference between POS and PTS and the pure frequentist statistics power of a study is the consideration of uncertainty. The frequentist power calculation typically assumes no uncertainty in treatment effect, while POS and the PTS, the Bayesian version of the power, accounts for not only the expected treatment effect, but also its uncertainty. Sometimes the prior signal, such as the ones from proof of concept study, may be relatively weak, or with a large variability. So POS and PTS could be very high as 99% or very low as 10%, despite a trial stated a statistical power of 90%. So in practical terms, considering POS and PTS is crucial for decision-making in phase 2B to phase 4 trials. These metrics guide our judgment and help us make informed choices based on projected outcomes and uncertainties. Next slide, please. So for the project at hand, to calculate the POS under average power or assurance, the success criteria, there are two. First is to achieving statistical superiority of the active group over a control group in a phase 3 study. And secondly, is the observed treatment difference is greater or equal to a certain criteria considering target profile. The average power is obtained by weighing the success probability at various observed modeled treatment difference D from the phase 2 study by the likelihood of the true treatment difference delta under the posterior distribution of delta given D. So as you can see from the formula is that the average power can be impacted by various factors. First is the location, that is the treatment difference of the prior distribution formed based on the phase 2 study has a large impact on the calculation of POS and PTS. And average power increases with increase in numbers of the subjects in the phase 2 study. As the larger the sample size, the greater the precision of the estimate and the less uncertainty of the treatment effect. As shown in the previous slide presented by Zhang, for the project at hand, the sample size of the phase 2 study is relatively small in the in stage 3 in the muscle cohort, with 9 patients from each treatment group, which restricts the traditional statistical approach of calculating average power to responder analysis, which is targeted as a moderate improvement in TIS, which is a binary endpoint. So as the point estimate can be markedly influenced by one responder difference. So in the next portion of the presentation, Zhang will share utilizing exposure response modeling and clinical trial simulation for evaluating probability of success.
Thank you, Min. So on the topic of using clinical trial simulation to determine probability of technical success, as Min mentioned, we use an exposure response model. Now, this wasn't a very simple exposure response model, so I will just sort of walk us through what that looked like. And just a very small amount of the sort of verification that was done with the trial data. So for the exposure response model, there were 11 responses that were all jointly modeled. Of these 11 responses, they included all of the TIS subscores, the continuous underlying score that's used to calculate the absolute percent improvement is what I'm referring to there. CDASI was also modeled, and then SF36 was found useful to include in the model. Each of these responses was driven by a PK/PD-based marker, that being percent of total interferon beta that was unbound. So that getting back to what was said earlier about it would be difficult to develop a PK/PD model, that is kind of a different story, but we were able to develop a nice predictive PK/PD model that did additionally predict the interferon beta data as that came in later, but was predictive of the endpoints that were available at the time as well. So as well as having all of the responses being driven by that similar exposure metric, we also shared information among the endpoints by sharing the same turnover rate in these indirect response models, the same placebo effect across all of the endpoints, and a kind of Emax adjustment to that percent of unbound interferon beta. Further information was shared by, like I said, the continuous representation of all of the responses were modeled, so their baselines were considered as being potentially correlated. So they were all captured, the baselines were captured with inter-individual variability in this large variance-covariance matrix where the off-diagonal elements were allowed to be fitted to greater than 0. So that sort of helped really kind of fill in the information on a small sample size. The approach here was that technically we were missing endpoints. So we were missing a lot of the TIS endpoints in our skin-predominant subjects. So kind of borrowing from the idea of full random effects modeling using that large variance-covariance matrix, if you're familiar. And so one of the VPCs that was done for this assessment was looking at how well TIS category was predicted at sort of the key time points of 4, 8, and 12 weeks. On the left, we have the placebo response, the placebo group, and on the right, we have the 600 milligram group. I do indicate the crossover there, but that happened after the bounds of this VPC and the sort of key analysis that was used for PTS. The bars that we see there and the numbers at the bottom of the bars, those are the actual responders at that level. And then the points and error bars are the median model predicted responder count, and then the 95% confidence interval of that prediction. So what we can see is that for essentially all of the categories and time points, we are within the bounds of the prediction, except for just the slightly higher response in placebo, which was kind of noted as being observed, where we had higher major responders than we predicted with the model at 12 weeks. But there's a lot of points where we are very close to the central tendency, which is also nice to see on occasion. And of course, this was not the only VPC that was contributed to support for the model. There were a number of other endpoints that it predicted very nicely. So ultimately, it was decided it was appropriate to use for PTS determination. So the methodology that was used here was a little bit different, although same sort of theoretical basis as what Min described, using clinical trial simulation, which is not necessarily new. Like it's been done before in the literature, but just to walk through how we did it. So for the clinical trial based PTS, that probability distribution of an effect size was driven by the random effects variability parameters. In the model, it was just inter-individual variability and residual variability, and then parameter uncertainty. So the actual uncertainty around the best fit parameters in the model. I use the same target response criteria that was used in the average power approach. So same thing to decide, where it counts as technical success. And then I also considered several penalties on the model predicted effect size. So the model, of course, predicts as an estimand what the effect size is. And that effect size for this case would be kind of what we would expect if we ran stage 3 of our phase 2 study 1,000 times. But then I applied various sort of penalty factors to that, and I'll mention in a second how it was applied. But there was a reduced version of the model-based effect size, which I call reduced model. There was one that was slightly above the observed effect size, which I call increased observed because it was slightly higher than the observed effect size. And then all the way at the bottom, 2/5 of the model predicted effect size was the observed effect size. It's important to note that 2/5 sounds like a big difference, but as Min mentioned, a difference of one responder one way or the other could have made a huge difference in our observed effect size. There's also very wide confidence intervals. And it's important to note that for the response we were looking at, the confidence interval from the observations contained the median of the simulated effect size, and then the confidence interval from that model-based effect size did contain our response. So it's kind of a nice little kind of internal consistency there with the model and the observation. So the actual methodology used for the clinical trial simulation, 1,000 trials were simulated where it was essentially matching the protocol of our phase 3 study. For each of these trials, like I said, the parameter when uncertainty was considered. So all the parameters used to generate data for each trial were different and sampled from the uncertainty, which is based on the sampling importance resampling. It was sampled from those parameters. And then R was assigned as either 1 or 0 if the delta TIS in the moderate response was greater than our target. That delta TIS observed in a simulated study, or calculated for a simulated study, whatever that was, right, which was some variability around the model-based effect size, was multiplied by these penalty factors to get at these reduced effect sizes. So, you know, if we observed 50, then what we used for our penalty, if we calculated from the simulation a delta of 50, then it was just multiplied by 2/5 to get what the penalized effect size is of 20. So that's sort of how that was done. Then if the target was was greater than 20, then the trial that was not penalized would get an R of 1, and the trial that was penalized would get an R of 0, right? So then the PTS estimates at each of these sort of penalty levels from clinical trial simulation were based on the probability of our trials meeting that criteria, or probability, or the percent of trials that had R equals 1. I'm sure there'll be some questions there, but hopefully I explained it clear enough. And those of you who have done something similar, I hope it resonated with what you've done. So then what were the results of MIN's average power approach based PTS and the PTS from clinical trial simulation, or PTS CTS? So in this slide, I recognize there's a lot of kind of abbreviations and a lot of kind of notes. I will kind of walk through what I'm showing you here in the table. There's a lot of kind of transformations to be on relative scales, just to kind of keep the direct information a little bit obscured. So on the left, we have the various effect sizes that I talked about, and then the label that I used before. So the reference effect size is the observed effect size, or ES OBS. Slightly higher than that would be the increased observed. Higher than that would be one that was slightly reduced from the model. And then at 2.5-fold the observed effect size, we have what was the model informed or the model predicted effect size. So in the next column is we have a PTS for each of these effect sizes from the average power approach, and we have a PTS from the clinical trial simulation approach. When you calculate those using the same effect size, it's interesting to see that you get a much lower PTS from clinical trial simulation at these lower effect sizes. But then even when we're talking about the model informed effect size, you only get about 1.5-fold the PTS using clinical trial simulation that you do from average power. So that is to say, from my end, it's not exploding out the PTS or looking overly optimistic on an effect size from an effect size to an effect size basis. But what's really interesting is if we weren't using modeling here at all and we didn't incorporate any other information, our PTS would have to be based on the observed effect size, and we would have to be using the average power approach. So what's really interesting is how do our various PTSs compare to our average power PTS at the observed effect size? And what you can see when we go all the way up to the model-informed effect size, we get about a 5-fold higher PTS using the average power approach at the model-informed effect size than we did at the observed effect size. Compared to the PTS using clinical trial simulation at these different effect sizes, you can see it's, you know, as we already saw in this column, we have a much lower clinical trial simulation PTS at the observed effect size. But then if we use clinical trial simulation without penalizing anything within the model, just assuming that the model is correct, then our model-informed effect size gives us a PTS that is sevenfold what we would have had as the average power PTS at the observed effect size. So, as I mentioned, we get a 5-fold increase just sticking with the average power approach and using the model-informed effect size. And it's interesting to note that depending on where your input effect size is compared to what your target effect size is, clinical trial simulation is either more conservative or more supportive than the average power PTS. But in some cases, it is basically going to give you the same answer, which is good to see. So, and if there's any questions about this, I can linger on this slide when we get to those questions. We've actually discussed a few different approaches. We didn't just discuss average power and clinical trial simulation. We addressed kind of augmentations to both of them. But what are the benefits of each approach? Well, there's what I'm going to refer to as traditional average power approach, which is where we use the effect size if we don't incorporate any other information, which you would do, in situations like this. But let's say you just use the observed effect size. Well, what's nice here is that the uncertainty of that observed effect size is already being taken into account when you are determining that PTS. So it's kind of already accounted for. So making adjustments for it, you have to really kind of defend that. And with the uncertainty, with the very wide distribution that we're talking about in this case, the average power approach is much less likely to say, well, you have a 0% probability of technical success if your input true effect size is less than the target difference. But then what we also considered was average power with a model-based effect size, or using a model to justify having a higher input true effect size. Well, what's nice about that is that it has all of the same benefits of the uncertainty as using average power with the observed effect size. But if there's a lot more information feeding into a higher effect size, you can kind of take advantage of that more information without really changing very much of the methodology. And then if that input effect size, true effect size, is higher than your target difference, average power is less likely than clinical trial simulation to be overly optimistic in its PTS estimation. And then CTS without penalty is just sort of the typical what you might do if you're doing PTS with an exposure response model. What's nice about that is you really have complete control over how the simulation is set up. You're able to consider early factors that have a effect later on in the longitudinal time course, and then you can consider other interesting things like, well, we have a very high probability. For example, we have a very high probability of meeting our actual primary endpoint, but we really think PTS should also consider this additional endpoint. So you can kind of consider joint PTS, which would be relevant if we had to consider safety. In this case, you ultimately, your PTS would be more of a joint PTS. You can also consider special statistical factors like skewed variability in a different way than you could in average power. And you can explore other factors like, what if our dropout was much higher than we expected? Or what if our effect was much higher in placebo than we expected, or something like that. But then you could also kind of do an empirical penalizing factor like was explored here, where let's say your model-based effect size is a little hard to swallow, it's a little too good to be true, then you can kind of just empirically change that, justifiable reasons like we did here. You can kind of just grade it on a scale from model-based to observed. And that might be an approach that could be considered without making very many changes to the model, just more based on processing the simulation results. So then before we wrap up and hand over for questions, just wanted to get to the conclusions and what suggestions we have from here. So obviously this is a very appropriate audience for this presentation because I think these results show that PTS does not have to be something you go to statistics for and statistics figures it out in a silo, or you go to Pharmacometrics for and they figure it out in a silo. It's best done when it's a collaboration between those two groups, and obviously other associated groups. If QSP is relevant or ClinPharm is going to particularly give good insight, even clinical, like it's much better if it comes out of a discussion rather than just one of these groups trying to carry that whole load. And then just kind of on the modeling front, clearly we only had a small amount of data for the endpoint that we were interested in. But our modeling considered the whole wealth of data, the totality of data that we had for the study, for the most part. And that provided a lot more information on this very small, lower information endpoint that was a lot more critical. And obviously that was very useful and something that should be tried if you have maybe broad data, but you know, maybe shallow in some respects. Then, of course, in this case, it was a rare disease. It's clear that it could provide benefit in rare diseases, but it could also be very useful early on as data is coming in to kind of use this approach. And that's it. I'll, of course, bump around the slideshow as questions come in, but I think we can open the floor to questions. And thank you.
Thank you both, John and Men, for this great presentation. I really appreciate both of you taking the time to present some of the good work you've been doing. And I'll just start going through some of the questions. First should just be the probability of technical success from clinical trial simulation gave both very low and very high relative estimates of the observed and model-based effect sizes. And then as you're saying, penalizing the effect size was proposed to correct for this, but what else could have been done to decrease the certainty of the estimates from clinical trial success?
Yeah, so as I mentioned, it was just kind of a fudge factor. We did consider parameter uncertainty, but obviously there were some parameters that were much more influential on the variability that was ultimately observed in the delta of the TIS response. Those being the kind of, you know, Emax parameter that sort of adjusts for the percent interferon bound, but then also the PK/PD parameters around that. So I think if the uncertainty around those parameters, again, this would kind of be an artificial thing, but if the uncertainty around those parameters was kind of inflated, so we had a broader range, that might be a kind of more statistical way of doing it, you know, because we had about 80 subjects in the trial, but all the parameters were fitted with the certainty kind of you would expect for 80 subjects, even the ones that were in kind of the lower information group. So kind of, you know, expanding out the uncertainty of the TIS related parameters, that would probably be one statistical way to handle it. So thank you.
And again, I'll remind everyone, we do have the Q&A, so please type in any questions you have. One of the questions is, has CDASI been correlated with physiological parameters? Is it mechanistic?
Yeah, so it was found to be correlated with, and I did briefly show a result here. So this was sort of associated with Pfizer study. So this was actually a publication that used a version of the gene signature score that we used, but it also directly measured interferon beta concentrations. It's really difficult to measure these concentrations in healthy or even less severe DM subjects, just because there's not particularly high sensitivity assays. I think what I saw, the lowest sensitivity you can get for interferon beta was around 1.2 picograms per mil. For our study, our LLQ was 10. And I believe it's clear from the data that, you know, most people will have less than 1.2 picograms per mil. So it has been correlated to interferon beta concentrations. It's been correlated to IP10, and it's been correlated to signature. Although I don't think there's like a QSP model necessarily that mechanistically links those observations to CDASI. That's work that would be interesting, but it's yet to be done as far as I know.
All right, and then I just want to remind everyone this will be posted on our website the recording of this presentation, so you can find it there if you would like. Another question we have is, what ways did you consider to explicitly bring in prior information, and how do you communicate that to the study team?
Yeah, so you might also have a perspective there, but for my modeling, there were 2 large phase 3 studies that had been conducted at the time. So we had placebo information from them. So, from those phase 3 trials, we were able to see what the average TIS response in placebo was, and then one of them even sort of categorized it into minimum, moderate, and major improvement. So I was able to simulate out large phase 3 trials and say, well, this is what our expectation is for placebo groups, and it's very consistent with the placebo group when summarized in the same way, because I think there's a bit of a skew to TIS. So when comparing the mean to the mean, we are very similar in our response placebo-wise to these large phase 3 studies. So that was influential in sort of the predictions of the model compared to what we observed in placebo. Min, do you have anything?
Yeah, I agree, Zhang. So I think the other thing maybe we also look into is the published data on various trials. Although in this rare disease setting, it's quite limited. So there are prior information we can get in terms of treatment effect on this particular disease is from a clinicaltrial.gov publications and also literature assertion.
So the next question is, how was the uncertainty of the PK/PD parameters determined? So was it determined explicitly based on specific data sets using the statistical model, or vaguely based on experience? And if in the latter case, would such experience be the sort of individual based and affect the PTS calculation?
Yeah, so for the PK/PD model, so not the exposure response model, in both cases, for the simulations as they were run, each subject had their own individual predicted, of the parameters that have an individual parameter. They were predicted for the subjects, and then RUV was applied to each observation. And that was true for both the exposure response model and the PK/PD model. The uncertainty of the parameter estimates there, as I alluded to, for both the exposure response simulation from the PK/PD model and the exposure response and the PK/PD model predictions themselves, the parameters were pulled from SIR-based resampling of the potential parameter estimates that would be associated with the model. So the same thing that was used to judge the parameter uncertainty, that was used to feed into the parameter estimates that were used for each simulation. I hope that addressed the question.
So the next would be, was average power calculated using a conjugate Bayesian prior model? So for instance, a normal for continuous TIS, and was the MC-MC simulation used? I believe this is for Min.
Mm-hmm.
Okay, so yeah, so for the average power calculation from a traditional approach is that the endpoint we're looking at is the moderate improvement in TIS as a binary endpoint. So for the calculation is that I think there's 3 options I know of. One is we can do the estimating, basically essentially is how to estimate the integral. So to estimate the integral, we can use the quad subroutine in the SAS-IML, so QUAD subroutine, to do that calculation. And also I'm aware of EAST also has a package to do a Bayesian power calculation, which with the EAST, it's more, the interface is more user-friendly, which does not require programming skill. And also simulation, say like with a Monte Carlo simulation can be used either with a SAS or a R package.
And for what it's worth, in the exposure response model, there was no average power type assessment that was done there, and MCMC was not used. It was all just from actual clinical trial simulations, and then just directly processing the data.
How did you evaluate the predictive performance of the model given the data set was very small? And did you split into the data into training and test sets? Did you use cross-validation? And I'll just add that the visual predictive checks may not be a familiar diagnostic to everyone on the call.
Yeah. So it was mainly through visual predictive checks here. So where we took the input data set for the model, and we simulated from that what we would expect to observe from the model 1,000 times, and then use, in this case, 1,000, and then summarize the observations based on what we were trying to see if we predicted well. So in this case, like I said, we could look at from the simulations and then from the observed, the responders at each category. It's not typical, and as you can imagine, for a 9 per arm study, it wouldn't be super feasible to do a kind of cross-validation approach. So the VPC approach was what was used, and then of course you could do a kind of numeric check on these same numbers. But along with looking at the category of TIS improvement for each of those subscores, as I showed in this slide, which might take a second to show up, each of those could also be broken down into categories. So we did look at categories of improvement or improvement score from each of these subscores, and all of those also predicted well, similar to the TIS. And then, like I mentioned, the model explicitly was modeling the continuous score that was underlying the calculation of absolute percentage improvement. Those predictions of the continuous score were looked at through that visual predictive check pipeline, I guess, and they predicted well. Of course, with only the 9 subjects per arm, we were just looking at the median prediction, but we were within the confidence intervals as expected from the model. And then for the wealth of data we had from stages 1 and 2, we also looked at those endpoints, CDASI. There are like 2 main breakdowns of CDASI, which we didn't cover, but those each were predicted well by the model. SF36, the parts of that were looked at were predicted well by the model. So it was mainly through VPCs, and then the optimization of the model was through just a gradient descent algorithm, FOCEI, for those familiar with that. Yeah, so that was sort of the validation and verification that was done. There was also things predicted by the model, like that subjects with muscle predominance would have lower baseline MMTs, which suggests worse muscle disease than subjects who are skin predominant, which also helps support the model because that's consistent with expectations. Skin predominant would be expected to have better baseline muscle score. And then, of course, I mentioned how we consider it with external data. So those all fed into support for the model.
I know we're at the hour, so I'll just have one more question. And I believe that one of them was this published, and I don't think you published it yet. But the last question will be, in terms of setting up the penalties, how does the current approach, shrinking the effect size, compare to the prior approach, shrinking the effective sample size? This may be for Min.
Yes.
So sorry, maybe I'm not quite understanding the question in terms of sample size for planning for the phase 3 study. Is that question? Sorry, could you repeat?
So it's in terms of setting up the penalties, how does the current approach with shrinking the effect size, say the 2/5, 3/5, 4/5, compare to a prior approach where you shrink the effective sample size?
So you know, I think we haven't necessarily explored in an exposure response clinical trial simulation framework. Sort of, if we're kind of building off my answer to an earlier question, where the uncertainty is adjusted for the lower sample size, we haven't done that comparison. But then this comparison that we have here is how, you know, 1-to-1 the sample size based kind of average power PTS compares to the clinical trial simulation. But beyond that, I don't know if I fully understood the question either. But yeah, that's sort of my response to that.
I apologize for people who ask questions that we haven't gotten to, but I know we're at a little over the hour, and so I don't want to take everyone's time. So I just want to thank our speakers, Min and John, for an excellent presentation. And we hope to have these joint webinars every twice a year is our goal at the moment. So we hope to see you next time. So thank you again, everyone, and I hope you have a great rest of your day.
Thank you.
Thanks.